Challenges and limitations. Despite its enormous potential, CRISPR-Cas9 also faces several challenges and limitations. Here are a few examples. Off-target effects. One of the major challenges of CRISPR-Cas9 is the potential for off-target effects, where the Cas9 protein cuts DNA at unintended locations. This can cause unintended genetic mutations and potentially lead to health problems. Delivery. Another challenge of CRISPR-Cas9 is delivering the gene editing machinery to the target cells or tissues. This can be particularly difficult for diseases that affect the brain or other hard-to-reach tissues. Immune response. There is also concern that the immune system may recognize the Cas9 protein as a foreign invader and mount an immune response, which could limit the effectiveness of the gene editing technology. Ethical concerns. As mentioned earlier, there are also significant ethical concerns surrounding the use of CRISPR-Cas9, particularly in the context of human germline editing and the potential for creating genetically modified organisms. Future directions. Despite these challenges, scientists continue to explore the potential of CRISPR-Cas9 and other gene editing technologies. Here are a few areas of future research. Improving precision. Researchers are working to improve the precision of CRISPR-Cas9 and reduce off-target effects. One approach is to use smaller guide RNAs that can more precisely target the desired DNA sequence. Developing new applications. Scientists are exploring new applications of CRISPR-Cas9, such as using it to study the function of different genes or engineer new materials. Addressing ethical concerns. Researchers and policymakers are working to address the ethical concerns surrounding CRISPR-Cas9, particularly in the context of human germline editing and the potential for creating genetically modified organisms. Developing delivery methods. Scientists are also working to develop new delivery methods for CRISPR-Cas9, such as using nanoparticles or viral vectors to deliver the gene editing machinery to target cells or tissues. Regulation and governance. As CRISPR-Cas9 and other gene editing technologies continue to advance, there is a growing need for regulation and governance. Here are a few examples of current regulatory frameworks and governance mechanisms. National regulations. Many countries have established regulations governing the use of gene editing technologies. For example, the United States has issued guidelines for the use of gene editing in research, while the European Union has issued guidelines for the ethical use of gene editing in humans. International governance. The International Summit on Human Gene Editing, which was held in 2015, brought together experts from around the world to discuss the ethics and governance of gene editing technologies. The World Health Organization is also working to establish guidelines for the responsible use of gene editing technologies. Professional guidelines. Professional organizations, such as the American Society of Gene and Cell Therapy and the International Society for Stem Cell Research, have also issued guidelines for the ethical use of gene editing technologies. Public engagement. There is also growing recognition of the need for public engagement and dialogue around the use of gene editing technologies. This includes efforts to educate the public about the science behind gene editing and to involve stakeholders in discussions about the ethical and social implications of these technologies. Public perception and communication. As with any new technology, public perception and communication are important considerations for the successful adoption and implementation of CRISPR-Cas9. Here are a few examples of challenges and opportunities related to public perception and communication. Public awareness. One of the challenges of CRISPR-Cas9 is that many members of the public may not be aware of the technology or may not fully understand its potential benefits and risks.